Hello, my name is Alina Montgomery. I am a recent graduate of the Laurentian Biomedical Biology program, and toward my undergrad, I completed a fourth-year thesis under the supervision of Dr. David McLean, which I will be sharing today, and it is entitled Investigating the Interaction Between Doxorubicin and Exercise on Nitric Oxide Metabolism in Heart and Liver Cells. In the next 10 or so minutes, my intentions are to discuss doxorubicin, its dose-dependent side effects, and the role of acute exercise and nitric oxide. I will be sharing my research study and some ideas for potential future research. In 2019, more than 220,000 Canadians were diagnosed of cancer, and of this population, many will receive doxorubicin as a part of their treatment. So doxorubicin, which is abbreviated to DOX here, is an anthracycline antibiotic, and the cytotoxic action of this drug is nonspecific, which results in widespread damage throughout the body. Its specific mechanisms of action include DNA intercalation, inhibition of DNA topoisomerase II, and free radical generation. Cancer treatment involving doxorubicin is not without its complications, and many of these side effects are dose-dependent, which limits the use of the drug. So, for example, DOX-induced congestive heart failure drastically increases from 1 to 2% with a cumulative dose of 300 milligrams per meter squared, to 20% as this dose surpasses 550 milligrams per meter squared. And then as many as 40% of people that have received this drug report liver injury to varying degrees. So this figure describes three commonly hypothesized mechanisms that contribute to this tissue damage. The first is the upregulation of nitric oxide synthase, shown here as NOS, and this is the enzyme that produces nitric oxide. So with an increase in its activity, there's an increase in nitric oxide production. And then the second and third are redox cycling and oxidative stress, both of which result in an increase in superoxides. And then with both nitric oxide and superoxide in excess, they readily bind with each other, which forms reactive species, which results to damage of surrounding tissues. Acute exercise offers multiple protective properties to try and mitigate this tissue damage. The first is the upregulation of antioxidant defenses, heat shock proteins, and organelle tolerance to DOX. And the second is the downregulation of apoptosis signaling and lipid peroxidation. And all of these work together to prevent the formation of reactive species, which will result in a decrease of nitric oxide levels. So the purpose of this study was to investigate the interaction between DOX and exercise on nitric oxide metabolism in heart and liver cells, and we hypothesized that acute exercise performed following the administration of DOX will lower nitric oxide levels in heart and liver tissues of the exercise rats compared to those that did not undergo any exercise protocols. The experimental design of this study was approved by Laurentian Animal Care Committee prior to it starting. There were 30 male Sprague Dawley rats used, and a total of 5 groups labeled A to E, and each group had 6 rats. Groups B, C, and D underwent a 2-week acclimatization protocol to the swimming exercise that was used, and that just involved placing the rats in water for 30 minutes with a weight of 4% of their body weight attached to their neck with a harness. They swam on days 3, 6, 9, and 12 of the 2-week period. On days 3 and 6, they could touch the bottom of the container for the whole exercise, and on days 9 and 12, they could only touch the bottom for 25 out of the 30 minutes, and then for that last 5 minutes, they swam unassisted. Groups A and E didn't participate in this protocol because they also didn't participate in any exercise during the experimental period. And then after the acclimatization, all 5 of the groups received an interperitoneal 4.5 milligram per kilogram dose of DOX, and then this injection marked the beginning of the experiment. This diagram shows the timeline of sacrifice and exercise protocols conducted over the 48-hour experiment, so beginning with all animals receiving DOX at 0 hours. And at 24 hours, Group A, which did not participate in any swimming, was sacrificed. Group B swam for an hour and was sacrificed immediately after, and then both Group C and D swam for 1 hour. And then at 48 hours, Group C was sacrificed. Group D swam for the second time and was sacrificed immediately following. And finally, Group E, which didn't participate in any swimming, was sacrificed. And I think here it is important to add that there is no control group in this experiment that didn't receive DOX because the experiment was originally designed to measure DOX in tissue after exercise. So a control in the original design involved the unnecessary use of animals. But in this current study, the unexercised group essentially serves as a control. Immediately following euthanasia, the hearts and livers were extracted from the animals and flash frozen. The hearts were sectioned into the 4 quadrants, and a small section of the liver was removed using a scalpel. We freeze-dried all of the samples for around 24 hours. We calculated a wet-to-dry ratio of the samples by weighing them before and after to ensure that we removed all of the moisture. And then 3 to 5 milligrams of the powdered samples was combined with 0.5 molar perchloric acid, and this was to extract the proteins from the tissue. We used the 2-ADI Sievers nitric oxide analyzer for our analysis. This system used vanadium chloride to convert the nitrates and nitrites in the tissue samples to nitric oxide. So the tissue PCA samples were injected into the system in duplicates, so we got accurate and consistent readings for each sample. And then the area under the peaks that were made from the injections was quantified using a standard curve that was constructed from 6 known concentrations of sodium nitrate. 3 statistical procedures were used to analyze the data. The average nitric oxide concentrations were calculated for groups A to E. Heart and liver data was tested separately for normality. Significance in the heart sections was tested using an ordinary one-way ANOVA, and post-hoc analysis of this ANOVA was a Tukey test, and that was used to identify differences between groups. And then for the liver, significance was tested using a Friedman test. Moving on to the results, this graph shows nitric oxide concentrations after the administration of DOX in liver tissue. So we found no significant differences between the groups, and as you can see, the concentration of DOX between the exercised and unexercised groups is quite similar. Group A had the highest concentration of about 14 micromole per milligram, compared to the lowest seen in Group B, which was about 12 micromole per milligram. So the lack of change we observed in the liver is likely due to the acute exercise protocol that we used. Because of this organ's lack of contractile ability during exercise, as well as its composition of mainly loose connective tissue, a longer exercise protocol was probably needed to develop mitochondria that are able to resist DOX-induced damage, as well as evoke changes in the reactive oxygen species pathways. This slide shows nitric oxide concentrations across all 4 chambers of the heart. We observe significant differences in the left atrium and the right ventricle. Starting with the left atrium on the far left of the screen, the significant difference is between groups D and both unexercised groups A and E. In this section, the unexercised groups had nitric oxide concentrations of about 20 micromole per milligram, and then the exercised group D had a decreased concentration of about 16 micromole per milligram. And then moving to the right ventricle at the far right of the screen, the differences were observed between groups A and group C. Group A, which was an unexercised group, had a concentration of about 15 micromole per milligram, and then the exercised group C had a concentration of about 10 micromole per milligram. We didn't see any significant differences in the remaining right atrium and left ventricle, and this might be due to the small sample size of 6 or the lack of control group, but they do follow a very similar pattern to the previously mentioned sections, showing a decrease in nitric oxide within the exercised groups compared to the unexercised ones. So why we saw these differences in the heart rather than in the liver can be attributed to its muscular composition and the higher density of mitochondria that's likely faster in developing a resistance to DOX compared to the mitochondria in the liver. Also, the heart's increased activity during exercise upregulates the cytoprotective proteins and decreases oxidative stress more than what we observed in the liver. In summary, the original hypothesis was partially supported by the results of this study. We observed that acute exercise was able to lower nitric oxide levels in cardiac tissue, but not in liver tissue. Further studies are warranted to better understand the role of exercise on nitric oxide metabolism in the liver, but these results found in this study do strongly suggest the protective role of acute exercise in mitigating cardiotoxicity that results from DOX treatment. Thank you everybody for your time and attention. and have a great day.